Hello everyone. In this introductory video, we will have an overview of engineering chemistry lab for this semester. So, in the engineering chemistry lab, we have several experiments planned for the semester. So, here the basic overview of this experiment you can see in this video. So, basically we need to have a manual for engineering chemistry lab, and in this current scenario one can download that manual from chemistry department website, BMS College of Engineering, and this is how it looks, the engineering chemistry manual. So here we are having total 10 experiments. So among them, the first 4 experiments are titration or volumetric analysis. In that, the first experiment we have is total hardness of the given water sample. Then we have percentage of copper in the DOS solution, brass sample. Then we have COD, the chemical oxygen demand of the wastewater. Then later, the fourth titration is percentage of iron in given rust sample. Okay. Then later we have instrumental methods of analysis. In that we have potentiometric estimation of ferrous ammonium sulfate using standard K2Cr2O7. And then we have pK of weak acid by using a pH meter. And then we have determination of calorific value of bomb calorimeter, conductometric estimation of weak and strong acid mixture, determination of copper by calorimetric method, and flame photometric estimation of sodium. So these are the 10 experiments currently we have for this semester, and for each experiment we will have a separate video wherein you can see the demonstration of conduction of each experiment. Okay. So in this today's video we will just see what the overview of the engineering chemistry lab and how to do. There is something called standard solution preparation using a standard flask or volumetric flask, that we will see in this current video. There are the 2 common apparatus used for volumetric analysis, that is for titration. So totally we have 4 titration. These are the common apparatus used in the titration. So the first thing, all of you are aware of this. This is a conical flask. Then we have a buret, then pipette of 2 different sizes. One is 10 ml, other one is 25 ml. Then we have a standard flask. Standard flask is used to prepare standard solution. It's also called volumetric flask. In this one can prepare standard solution of solute. Then of course you can have standard flask in different size. This is a 250 ml standard flask, and this is a 50 ml standard flask. Okay. So then how to handle this apparatus once you come inside the lab. So the first thing is that we have here a plastic bottle. It's called wash bottle. In this bottle always will be having always distilled water will be filled in this bottle. So you can use this for rinsing process. Okay. So once you arrive inside the laboratory, first thing is you have to wash all the apparatus. Whatever the apparatus you have collected against your name should be properly washed. So the washing means generally washing is done using tap water. Washing is using tap water. For example, so if it is buret, you can use tap water and thoroughly it can be washed. Okay, so the buret, the tip of the buret also should be washed, and you can properly wash all the parts of the buret. The same thing is true for conical flask as well as pipettes, and also for standard flask. That means whatever the apparatus is collected in the lab, the first thing is all of them should be washed in tap water. The first thing is wash them in tap water. After that, so all of the apparatus should be rinsed with distilled water. That is, now I have washed this with tap water. Tap water may be contaminated, so therefore it should be rinsed with distilled water. Okay, so I am using a little bit of distilled water and rinsing this. So you can avoid touching the chemical. You don't have to shake like this. You can hold like near the sink. You can hold like this and make sure that all part of the inside the buret is completely washed or rinsed in this case. So then you can discard the distilled water is present inside. So then this exactly same thing should be done for conical flask, it should be washed with tap water, then it should be rinsed with distilled water. It's also true for standard flask. Okay. Standard flask, you have a stopper here. You can close this lid. This is a stopper, so you can wash this with tap water. You can close this and properly wash. And then after that, it can be rinsed with distilled water. Okay. So now, so since the standard flask is washed with tap water, the funnel also should be washed with tap water, and it can be placed above standard flask. Now both standard flask and funnel is only washed with tap water. Now it should be rinsed with funnel as well as standard flask. You can see I am just adding water in such a way that the funnel is properly rinsed with the distilled water, and also you can rinse the standard flask. So then after placing this funnel over standard flask, one can go for weighing the corresponding salt for the given experiment. Today for demonstration purpose we are using potassium dichromate. The exactly same procedure should be followed for any other solution preparation. Okay. So today for demonstration purpose we will have a solution of potassium dichromate. We will see how to make a standard solution of potassium dichromate. We have potassium dichromate here. So I have taken a little bit of potassium dichromate in this weighing bottle. So this is a plastic bottle. Inside you can see there is orange colored substance or orange colored crystal, that is potassium dichromate. So here now you can see this is the weighing balance. This gives accurate weight up to 4 digits, 0.0000 up to 4 digits gives accurate weight up to 4 digits, and it's now in grams. So I am placing this. It is now tailed. That means the weight here is 0 gram. So I am placing this weighing bottle along with the potassium dichromate crystal inside. So the weight of the weighing bottle along with the potassium dichromate crystal is 6. So that is noted down. The weight of the weighing bottle plus potassium dichromate crystal is noted down. It is 6.3530 gram. So now we have weight of weighing bottle plus potassium dichromate. What I am going to do now is to transfer this potassium dichromate inside the standard flask. I am going to transfer this inside the standard flask. The care should be taken that you should not touch this weighing balance with wet hands. If you touch with wet hands, then there will be alteration of weight. Therefore, the hand should be dry when you are weighing. So now I have transferred this potassium dichromate inside and keep this aside. I will just reweigh this empty bottle. Reweigh the empty bottle. The weight of the empty bottle is noted down, that is 5.6756 gram. So we know now weight of the bottle plus potassium dichromate and weight of the empty bottle. If you subtract the weight of the bottle plus crystals minus weight of the empty bottle, you will get the total weight of the potassium dichromate transferred inside. Okay. So now we have to, so here if you see there are certain crystal there that is sticking to the funnel, so it has to be washed down. So you can add water. Now you can see here the funnel. In the funnel we have some amount of potassium dichromate sticking to the funnel. So it can be washed down now. So with using distilled water, I am using distilled water. So to make sure that there is no crystal that is sticking to the funnel, so we thoroughly wash it down. So you can see there is nothing inside. And also to make sure there may be some crystal sticking to the tip of the funnel. To avoid that, so you have to wash it from inside like this. Okay. So overall you have to make sure that there is no crystal that is wasted. Okay. Everything is now inside this standard flask, and now I am going to dissolve this carefully. So by gently shaking this solution and dissolving this. So I have added only approximately 30% of this. I have not filled completely, because if you fill up to here completely, to fill this volumetric flask completely, then mixing will not be possible, and the dissolution of solute will becomes difficult. Therefore, you should not add full solvent now. The solute should be dissolved first. So potassium dichromate is quite soluble in water, therefore it dissolves readily. The other thing to note down here is I am not using stopper now. So without using stopper, I am gently shaking this. So now you can see all the crystals of potassium dichromate inside this is dissolved. It's completely dissolved. So now I have to add the water to up to this mark. So this is a 250 ml standard flask. A 250 ml standard flask. The total volume is 250 ml if you fill up to this mark. Okay, this mark. So it has to be, the solution should be filled up to this mark. So now we have added only a little bit of water. So rest can be filled now. Once after dissolution of the solute, rest of the water can be filled slowly and carefully. So using this wash bottle, here we have distilled water. That means what I am filling inside this is distilled water. The care should be taken to avoid the crossing of this 250 ml mark. So if you cross this 250 ml mark, then we have to discard and restart the process. Therefore, should be careful. The addition of water should be done very carefully. So therefore, in order to avoid those things, what I am going to do is I will stop at this point. Stop at this point. So now I can comfortably fill up to this point using wash bottle. And later I have little control in wash bottle. I don't have a proper control in wash bottle. Therefore, what I will do is, in a clean, washed and rinsed buret, I am going to take distilled water. Okay. And I will add distilled water slowly. So here we have now distilled water. I am going to add distilled water inside this very slowly and carefully. So in the buret we have proper control. I can add slowly, drop by drop. So therefore I am using buret, and also the filling of this region can also be achieved using pipette. In pipette also one can take distilled water and add slowly, drop by drop. But the buret will afford you more better control. So therefore I am using buret in this case. So now in the buret you can see I am just adding very slowly, drop by drop. Now you can increase the rate of addition little bit. When it comes closer to the mark, you have to reduce the rate of addition. So you have to make sure that the lower meniscus of the solution is exactly coinciding with the line here. The 250 ml mark you can see here. It's the lower meniscus. Now you may need 1 or 2 drops. I am going to add 1 drop and check. So 1 more drop. Okay, so now you can see the lower meniscus is exactly coinciding with the 250 ml mark in the standard flask. Okay. So we are seeing lower meniscus, even though it is a colored solution, the intensity of color is very less, it is very lightly colored. As long as you can see lower meniscus, you can go ahead with the lower meniscus. So here, and the other thing is, while reading this, or while reading burette reading, or while measuring the solution in pipette, you have to keep to your eye level. The eye level and this mark should be at the same level. Okay, so to avoid any parallax error, if I see from here, it looks like the solution is below the mark, and if I see from here, it looks like above the mark. So to avoid this parallax error, you have to keep exactly in line with your eyes. So it is exactly now. The total solution is exactly 250 ml. So what the care should be taken here is, one, the addition of the last portion of the standard flask should be done carefully using a burette. Okay, and you have to avoid parallax error. Therefore, you have to see the lower meniscus of the solution at your eye level. Okay, these are the two errors that generally then it should be avoided. So if it crosses this 250 ml mark, then you have to repeat the process. Therefore, you should be very careful in this process. So now the solution is ready, but the thing is, here the solution, there is a concentration gradient. Means the lower portion of the solution is having more potassium dichromate concentration, upper portion will be having less potassium dichromate concentration. So now I have to have a homogeneous solution. To make homogeneous solution, it should be thoroughly shaken, thoroughly mixed. How to do it? It should be the solution. We have to use this stopper properly and close this properly and invert the standard flask. You get some gap here, so then it should be thoroughly mixed. Okay, so then allow it to go down, allow the solution to go down, and once again repeat the process. So it has to be done at least some 4 to 5 times to get a clear homogeneous solution. So now we have a standard potassium dichromate solution ready here. Okay. So now how to fill this, fill the burette using the potassium dichromate. How to fill? You see, I have filled here the distilled water. I am going to discard this distilled water. So now the burette should be rinsed with the solution to be taken. The first point is, all the apparatus should be washed with the tap water, then rinsed with the distilled water. It is true for all the apparatus in the lab. And the rinsing with the solution to be taken. In case of burette, it should be rinsed with the solution to be taken. And while filling burette, you cannot directly fill. You can use a funnel like this. And while rinsing, you should also rinse funnel. So just like so, I am rinsing both funnel as well as burette now. The funnel is also rinsed. Now the burette should be rinsed. Once again, I am not touching the solution with the hand. So here I am just holding like this. So all region of burette in this portion, everything is rinsed. Only here the tip of the burette should be rinsed now. Okay, that is also done. Now you can discard the solution. Now you can refill the burette. Fill the burette with the solution to be taken. am taking slightly more than 0 because I have to remove this air bubble. This region is empty now, the tip of the buret is empty. So open once fully. So if any air bubbles here will be going down, so then you can adjust to 0. So as in the previous case, the lower meniscus should be exactly coinciding with this 0 ml mark. The lower meniscus of the solution should be exactly coinciding with the 0 ml marks, and while reading it should be at your eye level to avoid any parallax error. So this is about the buret, and also you have seen how to wash and use a standard flask, how to prepare solution in the standard flask. And while using pipette, pipette first should be washed with tap water. Okay, first it should be washed with tap water. I am going to demonstrate the 10 ml pipette. I am going to demonstrate 10 ml pipette. Exactly same thing can be followed for 25 ml pipette. So it can be washed in tap water like this. And should be rinsed with distilled water. So while rinsing the pipette, you can take distilled water in a washed clean conical flask. Here I am taking distilled water. You should not immerse anything inside the wash bottle. The pipette may be contaminated and whole solution or whole distilled water in the wash bottle get contaminated. Therefore, the distilled water should be taken in a clean conical flask. Then, using this pipette bulb or pipette filler, I am going to wash this. So here, if you see pipette bulb, this is used to refill pipette. Okay, so how to perform this? Just press this region and immerse in the solution to be taken, in this case distilled water. And if you press this region here, then there is a suction and it will fill the pipette. You can see there is the water level in the pipette is increasing. So since I am just rinsing, I will stop at this stage. Then and remove this. So just all region inside the pipette is completely rinsed with the distilled water. In a similar fashion, one can pipette out any solution. The solution can be may be given in any flask. So you can use this pipette and a pipette filler, and you can pipette out using this pipette filler. Okay. So this is about 10 ml pipette. Exactly same thing can be followed for 25 ml pipette. And the important thing here in case of pipette, the rinsing should be done with distilled water. Also, rinsing should be done with a solution to be taken. Let's say I am taking potassium dichromate in pipette. In that case, it should be rinsed with potassium dichromate also. Very similar to buret. The important thing here in case of conical flask, the conical flask should be washed with tap water and rinsed with distilled water, but conical flask should never be rinsed with solution to be taken. This is the change in case of conical flask. Buret, wash with tap water, rinse with distilled water, rinse with solution to be taken. It's also true for pipette. But in case of conical flask, it should be washed with tap water, rinsed with distilled water and stop there. It should not be rinsed with solution to be taken. What happens if I rinse conical flask with the solution to be taken? There will be some portion of the solution. If I rinse with solution to be taken, you can see there are some droplets, right? Here you can see some droplet. That means some extra solution getting added into the conical flask. Therefore, to avoid that, the conical flask should never be rinsed. It should be only rinsed with distilled water, not with the solution to be taken. Okay? So the molarity of the standard solution prepared can be calculated using this formula. So we have used it today. The weight of the bottle plus potassium dichromate crystal was 6.3530 gram. And weight of the empty bottle was found to be 5.6756 gram. And therefore the weight of the potassium dichromate crystal that is transferred inside the standard flask is 0.6774 gram. So this is the amount of potassium dichromate transferred inside the standard flask, and therefore the normality of potassium dichromate equal to weight of the dichromate transferred inside divided by equivalent weight of potassium dichromate into 1000 divided by 250, because we prepared solution in 250 ml standard flask. And so if you substitute the weight of the potassium dichromate transfer, that is 0.6764 gram, and equivalent weight of potassium dichromate is 49. So the normality of the solution we prepared today turns out to be 0.0552 nanometer.